हेलो वेलकम बैक टू ऑल टू माय चैनल। आज हम पढ़ेंगे क्लास 11th का चैप्टर 10 दैट इज़ सेल साइकिल एंड सेल डिवीजन। इसमें हम पढ़ेंगे सेल साइकिल, एम फेज, सिग्निफिकेंस ऑफ माइटोसिस, मियोसिस, सिग्निफिकेंस ऑफ मियोसिस। तो शुरू करते हैं। Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organism. Growth and reproduction are characteristics of cell, indeed of all living organism. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. This newly formed daughter cell can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycle of growth and division allow a single cell to form a structure consisting of millions of cells. Cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also take place. All these processes, cell division, DNA replication, cell growth, hence, have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other constituent of the cell, and eventually divides into two daughter cells is termed cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosome DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. As you can see in the diagram, G0, G1, S, G2, साइटोकाइनेसिस यहाँ से शुरू होता है प्रोफेज, मेटाफेज, एनाफेज, टीलोफेज, साइटोकाइनेसिस, देन G1, S, G2 ये चलता रहता है और इसे हम एम फेज कहते हैं जो मैंने पढ़ा था. A typical eukaryotic cell cycle is illustrated by human cell in culture. These cells divide once in approx every 24 hours. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in about 90 minutes. The cell cycle is divided into two basic phases. हमारे में क्या होता है 24 घंटे का होता है और yeast में 90 minutes. The cell cycle is divided into two phases, interphase and second M phase. ठीक है, so what is interphase? What is M phase? M phase represents the phase when the actual cell division or mitosis occur, and the interphase represents the phase between two successive M phases. Okay, interphase दो successive M phases और M phase होता है actual जो mitosis होता है. It is significant to note that in the 24-hour average duration of cell cycle of a human cell, cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of the cell cycle. M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, karyokinesis, and usually end with division of cytoplasm, cytokinesis. The interphase, though called the resting phase, is a time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase divided into three further phases. G1 phase, gap 1, S phase, synthesis, G2 phase, gap 2. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S or synthesis phase marks the period during which DNA synthesizes or replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell had diploid or 2n number of chromosome at G1, even after S phase, the number of chromosomes remain the same, that is 2n. In animal cells during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Some cells in adult animals do not appear to exhibit division, that is, heart cell, and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not divide further exit G1 phase to enter an inactive stage called quiescent state G0 of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of organism. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this where haploid cells divide by mitosis. For example, Maine honeybees. Against this, the plants can show mitotic division in both haploid and diploid cells. From your recollection of example of alternation of generation in plants, chapter 3, identify plant species and stages at which mitosis is seen in haploid cells. M phase. Now let's see यहाँ boxes में क्या लिखा है. How do plants and animals continue to grow all their lives? How do cells in a plant divide by this time? Do you think all cells continue to divide in all plants and animals? Can you tell the name and location of tissues having cells that divide all their life in higher plants? Do animals similar meriphatic tissue? Some cells, organ phase. This is the most dramatic period of cell cycle involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cells is the same, it is also called as equational division. Though for convenience, mitosis has been divided into 4 stages of nuclear division, karyokinesis. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Though for convenience, mitosis has been divided into 4 stages of nuclear division, karyokinesis. It is very essential to understand that cell division is progressive process and very clear-cut line cannot be drawn between various stages. Karyokinesis involves following 4 stages: prophase, metaphase, anaphase, telophase. Prophase, which is the first stage of karyokinesis mitosis, follows the S and G2 phase of interphase. In the S and G2 phases, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centrosome, which had undergone duplication during S phase of interphase, now begins to move towards opposite poles of the cell. The completion of prophase can thus be marked by following characteristics event. Chromosome material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of 2 chromatids attached together at the centromere. Centrosome, which had undergone duplication during interphase, begins to move toward opposite poles of the cell. Each centrosome radiates out microtubules called esters. The 2 esters, together with spindle fiber, form mitotic apparatus. As you can see, cells in the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus, and the nuclear envelope. Metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosome is completed and they can be observed clearly under the microscope. This then is the stage at which morphology of chromosomes is most easily studied. At this stage, metaphase chromosome is made up of 2 sister chromatids which are held together by the centromere. Small disc-shaped structure at the surface of the centromere are called kinetochores. These structures serve as the site of attachment of spindle fibers to the chromosome that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to spindle fiber from one pole and its sister chromatid connected to its kinetochore to spindle fiber from the opposite pole. The plane of alignment of the chromosome at metaphase is referred to as metaphase plate. The key features of metaphase are spindle fibers attached to kinetochores of chromosomes. Chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fibers to both places to both poles. Anaphase. At the onset of anaphase, each chromosome arranged at the metaphase place is split simultaneously, and the two daughter chromatids, now referred to as daughter chromosome of future daughter nuclei, begin their migration towards the two opposite poles.
As new chromosomes moves away